big room. Great to be here. Thank you so much for coming and for your interest and concern about these complicated but important questions. I want to say that John just gave the take-home points for my talk, so I'm going to go sit down. Really, what I would like to do is to give you a way of thinking in more detail, but basically what John just finished saying. How do we think about what autism is in a way that makes sense of this process of resetting the metabolism, removing the stressors? How do you come to that? Now, I started out in autism research because I was doing brain imaging research, and I got handed a huge database of brain MRIs with accompanying data, and I was told, analyze this data and let's find the parts of the brain that match the test data. And I did that for some years and it didn't work, because that's what happened. Was I realized, that's not the problem. There's another problem going on. And so what I'm going to be bringing to you is a result of some years of thinking through what the data do tell us, and also what I've experienced in my small but regular clinical practice, having in my face the full-bodied medical problems of autistic children. What I'm going to do is explain to you that there is scientific plausibility to autism treatment. And this has to do with what many of us consider the emergence of a new model of autism. Now, when you have a new model, you have to shift your thinking, and I have this creature here, and the question is it a rabbit or a duck? There's a nose, this is a nose, or is it the back of the head? Is this the beak or the ears? So what I'm asking you to do is relax your preconceptions and understand that you can shift between models in the way you look at things. You also need to understand that everyone organizes their thinking with models, and when you go to the doctor is going to give you the advice on the basis of models. People using an older model are not always aware that there's a newer model. So they may not have studied it. They may not have thought it through. It's often dismissed without people having read the literature. So I just think it's important to understand that a lot of the roadblocks and resistances you run into are based on running into a model that doesn't yet encompass the things that you've started to think about. Now, this is a summary of the model. The older model would say that autism is genetically determined. The newer model would say that autism is environmentally triggered and genetically influenced. The old model would say that autism is brain-based, and the new model would say it's both brain and body. The old model would say that the brain changes are hardwired. The new model would say that metabolic changes, chemical changes, circulating factors play a big role. Oops. The old model would say that it's treatable but not curable, and the new model would say that it's treatable, and as John said, recovery is possible. So the question is autism a brain disorder, or is it a disorder that affects the brain? Now, I just wrote an article called, Is Autism a Brain Disorder or a Disorder That Affects the Brain? And I talked to Steve Edelson yesterday, and when my presentation will be posted on the DAN or ARI website, the article will be there along with it. So that article will take what I'm saying here, and it will give you 270 footnotes to support what I'm saying. Now, this is a very important point, and I think some of my psychology colleagues don't think about this very much, but the brain is wet. It's metabolic. It's an organ of your body, and it's also attached to the body. This is actually the shortest summary of what we're trying to say, that behaviors come from a wet brain, and the metabolism of that wet brain exists in the context of everything else that's going on in the body. So if you just get that, that's great. Let me try and flesh that out, so to speak. Now, I'm going to be talking about plausibility, and you need to understand that in medicine, now we talk about evidence, and I'm going to give you evidence, but there's a specific, evidence is sort of generic, and then there's this thing called evidence-based medicine. And evidence-based medicine is a kind of a brand name way of framing evidence. And evidence-based medicine says that you need to have double-blind, placebo-controlled studies in order to say something is true or not. And in that sense, there's very little evidence for what you're going to hear about in this conference. There are a lot of reasons for that, and one of them is that there's some pretty big problems with the whole way that evidence-based medicine is set up, and it's not just people in rooms like this who are critical of it, but it's causing problems in other areas. And I will get back to that. Particularly, one of the problems is that in 21st century medicine, we're learning about genetic individuality. We're learning how to characterize genetic individuality. You've probably heard in the news pharmacogenomics, pharmacogenetics, individual profiling. Evidence-based medicine lumps a lot of things together and differences get washed out. So we need to be thinking through not only the different evidence, but how to study it so that the genetic individuality is honored in an appropriate scientific way. And we want to have scientific resources allocating to studying the new model and allocated to developing rigorous methods that will allow us to study these things with the genetic individuality taken into account. Now, I want to go back to basics. Autism is defined in DSM-4, the Psychiatric Diagnostic Manual, by 3 behavioral characteristics. There is no biology in the definition of autism. It's all behavioral. Nobody knows the biology of autism, presumably because there is no one biology of autism. Biology is not part of the definition, and it's not part of there's no prognosis in there. When people hear about recovery, and they have their mind in the old model, they say, well, it couldn't have been autism, because they think that the incurability is part of the definition. That's complete nonsense. There is nothing about prognosis in the definition. If you have sufficiently strong presentations in the 3 behavioral domains by age 3, you meet criteria for autism, period. That's it. If you get better for whatever reason, you may no longer meet those criteria later on, but you met them at one point, period. That's it. So then it should be an open question about what are the biological underpinnings of what you had in the first place that allowed you to get over it. And I think this should be a question. It needs to be an open question. Now, I've listed here secondary features of autism: seizures, sensory motor, immune, GI, food. These are not part of the definition. They are frequent fellow travelers. They suggest strongly, as do many other things like brain findings, that this is a biological disorder, but we do not have a test at this time. No blood test, no brain test, no anything test that's wet or imaging, other than the behavior, to say you have it or you don't have it. So this is just all important to have as basic, clear understanding in your mind. Now, what are the arguments for claiming autism is incurable? Well, John already reviewed some of them. Genetics. Well, people assume that if there are genes involved, that means it's inevitable. That it has been thought to happen in utero, prenatally, and therefore it's fixed. I'm going to take these apart in the next slide, but let me just tell you what the arguments are. That it's hardwired, and anything that's hardwired in your brain is unchangeable, and therefore it's hopeless. So that's pretty much the line of thought that's used to dismiss anybody who would think that anything they could do biomedically could do more than control difficult behaviors. So what I want to make clear to you is that the evidence for the idea that it's incurable is weak, that treatment and recovery are plausible, and that there is evidence for that. So first of all, it's genetic. Well, John already said it's genes and environment. There's vulnerabilities that get triggered. That genetics can mean vulnerability and not fate. Second of all, brain changes happen before birth. Well, that's actually been pretty well refuted. Not that it doesn't happen before birth in some cases, but what's been refuted, and I'll go into it in a little bit more detail, is that we are seeing a lot of things happen in the brain after birth. So it's not a purely prenatal thing. Thirdly, well, if something's wrong with the brain, then it's a fixed thing. Well, we're finding, as Dave Weldon pointed out, that there's inflammation, there's oxidative stress. That's not wiring, that's physical illness. And that there's a brain-body connection. And finally, that nobody gets better. Well, there's a place, if your kid is getting better and you haven't signed up, I think, do I have it right, or are there hyphens, or www.autismistreatable.com is the ARI website where you can sign up and report the recovery of your child. And if you have a child who's recovering or recovered, I encourage you to use that website. Now, the old modular determinist model is a strongly genetic brain-based modular model that says that genes cause something in the brain and that causes the behavior. Well, that model is coming apart. Where are the genes and why haven't they been found? What about the environment? How do you get from gene to brain? What about the body? How can it be all hardwired if some kids get better? And also, it's not just a behavioral problem. So what I'm going to show you is an old model and a new model. Now, in the old model, like I just said, it's gene, brain, behavior. What I'm proposing is that instead of saying gene, we say pathogenesis. So that includes all different kinds of things, genes, environment, and complex interactions that occur during early development that can be considered causative. It's more inclusive. Gene-brain behavior is a special case. It's way narrow. We're looking to be more inclusive. Now, instead of saying brain, let's look at what is the brain? Well, there's molecular and cellular mechanisms. There's the tissue, and there's the way the information processing occurs in the brain. So you take that apart and you look at those, and the reason you do that is that those, breaking it down like that, that's, I think, where biomedical treatments target. These changes in molecules, cells, tissues, and processing, that's where you get what we see in the kids. The behaviors, the language problems, the cognition problems, sensory motor, body illness are all based on these underlying biological changes. And we need to have much more attention to the biology of autism. Another way of looking at it is that there are underlying mechanisms, and I'll talk about a few, and you're going to hear about a lot of this a lot more in the course of the conference, that can lead to brain changes in structure, brain changes in function, and body changes. And these mechanisms are influenced by genes and they're influenced by environment. But one set of underlying mechanisms can drive changes in many systems. If you look at it that way, and if you treat the underlying mechanisms, that can be very powerful. If you go after all the symptoms, you get this problem, which is in this joke, which is you may believe you've been overcharged, but remember, you're over-medicated. So you get one pill for the ear infections and one pill for the GE reflex and one pill for the behavior. Maybe if you treated the inflammation and the oxidative stress and the toxic problems and in the infection, which you can do in a different kind of way, you could overcome the symptoms in multiple domains. So now I'm going to review the claims that I just went through in a little bit more detail. The old model claim is that autism is basically genetic. Well, is it really all genetic? And if it is, then how do you explain the higher numbers? And what about the environment? So this is a slide about twin studies, and I won't review it in detail, but I will make a couple of key points. First of all, the argument about autism, the twin study supporting autism, is that if the identical monozygotic twins, if one has it, the other one has it 90% of the time. Well, that's not true. The data actually say that if one has it, the other one will have features of autism 90% of the time. If one has full autism, the other one will have full autism 60% of the time. So something else is going on. Second of all, these twin studies were all done none of them were done after the mid-1980s, and the numbers didn't start going up until after the mid-1980s. So we don't even have data on what this kind of question would look like if we studied it now. So it may not even apply to the biology of the kids who are showing up autistic now. So, and finally, there are other ways, such as in utero shared exposures, that can cause common outcomes in children. There's a slide that keeps disappearing on me, and I don't understand why, so I'm going to get out and show it to you in this other mode. It's the second time that happened to me. How totally bizarre. It was a slide of the California, it's blank, it's very odd. It was a slide of the California data from showing I'll just do it with my, it's not there.
I see that. Keep talking, it's okay, don't mind
Yeah,
me.
fine.
No, it's definitely not there.
It's not there, but it's the second time that slide disappeared on me. There's some
There's only
gremlin.
There's only one common element.
I'm going to make the slide again. Yeah. Okay, I'm going to do it with my hands. I hope you guys can see me. So basically, there's a graph, and it goes 1994 to 2002, and there's cerebral palsy, epilepsy, and mental retardation. They're kind of flat, and autism is zooming up to the sky, 273% increase. There's all kinds of caveats about this. It's like, well, it isn't necessarily that you can really infer rates from that data, but this is the kids who are getting diagnosed in California in the various districts that they have, centers. So then people said, oh, well, they were just diagnosing these people because the people who were mentally retarded were just getting relabeled, and really nothing has really changed. It's just that we have more awareness. Well, that was published, and then it was challenged, and then that was retracted. So now even some of the more conservative people are saying, look, we have to take some of these increases seriously. Let me just go on. Let me go back to here, and okay, back in business. This is Texas. This is the first of a series of studies, the rest of which are in progress, by Ray Palmer, Claudia Miller, and others at the University of Texas, on the potential association between autism rates, environmental mercury, and other toxins in Texas. So the first thing they did was they looked at autism rates by school district, and the darker the color, the more the autism. So you see that from 1990 to 1993, 1993 to 98 to 2000, there's a whole lot more dark blue in this picture. Well, keeping in mind this second one here, the second one here is now this picture. And if you look at it, then this slide down here is the federal toxic release inventory data from Texas. What toxins are released? Interestingly, the administration is trying to change the threshold. So instead of having to report 500 pounds of toxins, you won't have to report it till it's 5,000. So we'd have a lot less information, and this is information I think we would all like to have. Also, total toxicity. Uncanny overlap between these maps. The likelihood that this could be an entire coincidence. It may not be precise. They are working on methodological refinements, but it's a very strong correlation. And interestingly, look at this district, which has no toxic overlay in either of the other two maps. It turns out that this is the site of the oldest mercury mine in the United States. Now, on average, for each 1,000 pound of environmentally released mercury, and we're not talking thimerosal here, we're talking environmentally released mercury, there was a 43% increase in the rate of special education services and a 61% increase in the rate of autism. So this is under further study, but it's very provocative. So what do? Even my colleagues who don't believe that there's been any rate changes, and there are still some of these, who don't believe in the epidemic will still say, gee, there's a lot of kids. But if there's any debate and uncertainty in a major public health issue like this, what are the responsible things to do? Do you assume that nothing has happened until it's unequivocally proven? I think that's irresponsible, frankly. I think we need to include environmentally informed hypotheses centrally in research and policy. We need to look for environmental cause. We also need to look for the mechanisms. How does the environment, whatever the environmental factors are, impact the body? That's where we're going to help the kids. That's where the rubber hits the road, and that's where we can find treatment targets. So I think that that's what the implications are, even while we're not 100% sure exactly what caused it and exactly what combinations and exactly how many. We don't need to know that there's a problem we need to address. So what does genetic mean? Does it mean inevitability or vulnerability? This is a picture from a paper a few years ago showing all the places on the genome at that time that look like there may be genetic contributors to autism. It's all over the place. There's no genes of strong effect that seem to be common to all cases of autism. As of a few years ago from genome scans, significant would be above the red line. Most of the genes that were identified were far below that. So most of the genes have low levels of significance. Now, this is a slide from Kevin Becker, who works at the NIH, and he overlapped the significant areas in autoimmune disease, autism, and Tourette's, and he found a great deal of overlap. You can download this paper. I think it should be in your handout. And the point is that some of the genetic vulnerabilities aren't even specific for autism. They're more generic vulnerabilities. So do we have genetic determination or genetic influence? Steve Hyman, when he was head of the NIMH, he's now a provost at Harvard, said the following, and I'll read it to you. Gone is the notion that there is a single gene that causes any mental disorder or determines any behavioral variant. The concept of a causative gene has been replaced by that of genetic complexity, in which multiple genes act in concert with non-genetic factors to produce a risk of mental disorder. So that's the state of the science, and if you have people telling you we need to find the gene that causes it, they're not up to the edge, they're behind the curve. Now, so what does it mean to talk about genetic vulnerability? This is a slide, and I'll talk you through it. So this is a normal distribution, and the middle one is sort of the wild type, people who metabolize at an average rate. What your genes do to a significant degree is they make enzymes which drive chemical reactions. They also make structural proteins and other things, but let's talk about the enzymes. You can have a change in your genes that makes you metabolize things slowly, or you can have a change in your genes that makes you go fast. So if you burn up necessary nutrients too fast, or if you get rid of toxins too slowly, that's a problem. If you have both of those, it's a bigger problem, and then if you get hit with one of the stressors that you handled badly, that's a really big problem. So that pretty much is what genetic vulnerability is about. And also, there's an article by Bruce Ames that high-dose vitamin therapy can be used to drive the speed of slow enzymes. And that's a lot of what is being recommended here. Now, the NIEHS, the National Institute of Environmental Health Sciences, has an environmental genome project. I got really interested in this because I kept on reading gene reviews and finding they were only talking about brains, and I found that most of my genetics colleagues had never heard of the Environmental Genome Project, and I thought that was pretty interesting. Environmentally responsive genes include genes that govern many important things about how we function: cell cycle, cell division, I'm just going to pick out highlights, metabolism, immune inflammatory responses, oxidative metabolism and stress, hormones, signals, membranes, drug resistance. So all of these things have genetic influences because they're all biological, and the chemical way that you do that has a genetic component. These are environmentally responsive genes. So I got interested, I got a bunch of people together, I said, let's take the Environmental Genome Project and overlay it on top of the autism hotspots on the gene, and I found that there were a whole lot of genes in the Environmental Genome Project that nobody ever thought of as interesting. Why? Because they're mostly looking for brain genes. This paper just got accepted last week at Neurotoxicology. It's online now, it's not in print yet. But it's making the point that we need to think about gene-environment interactions in the way we study genetics. Why am I having this? Very weird. I have no idea what that one was that's out. But some gremlin got into the computer. So that's very scientific explanation. Let's see here. So geneticists, there was a recent autism genetics conference a couple of weeks ago, 3-4 weeks ago, maybe less, at Cold Spring Harbor Laboratories, and the geneticists were, oh my God, we need more phenotypic data. We need to know about the kids. Wow. But they're still just looking at behavior. They haven't figured out, and somebody did get up in front of them and said, we want wet measures. We want blood tests. We want urine tests. We want brain imaging, not just behavior tests that's way subjective. We need genetics to be related to autism biology. Biology is a critical point here. So here's a take-home point, going back to the pathogenesis mechanism phenotype, which I don't know, I hope you can read it across the room, but maybe not. Do genes cause autism, or do they create vulnerabilities to second hits from the environment without which some children might not develop autism? And this is what John said to you before. Next claim, the changes happen before birth, so they're hardwired and genetically determined. Is this true? What about the many changes that take place in autism after birth? And so, where do we get this idea that it's prenatal? Well, there have been a few neuropathological studies, analyses of brain tissue from people who had autism and died, that found changes in cerebellum, limbic system, and brainstem, suggesting that you could interpret as consistent with prenatal changes. However, a lot of neuropathologists, number one, a lot of neuropathologists never bought that. Number two, there are other interpretations. And number three, Dr. Bowman and Dr. Kemper went back with a new set of 11 or 12 brains that I've personally imaged in an MRI scanner and did not replicate those findings. They found that some of those findings Some of autistic kids did not have those findings, and some control kids did. So those findings are actually can no longer be construed as specific. So basically, we don't really know what's going on. There is data that prenatal and perinatal infection can lead to some of the changes we see postnatally. All in all, we have evidence that can be, which is strong enough to say that some cases of autism may have prenatal onset, but we don't have enough evidence that prove that all cases of autism are entirely prenatal. Very few people believe that anymore, and I just think you need to know that. I've had major people in multiple places tell me that that's over. Now, there's a lot of evidence supporting brain changes after birth. First of all, behavioral regression, the children who seem more or less fine and then lose it somewhere between 15 or 16 and 22 to 24 months. There was finally papers coming out this year, like analyzing home videos of parents gave, showing the kid was doing virtually fine, maybe minor problems, and then is gone. Now, is this, as my colleagues have sometimes tried to say, the unfolding of an inborn genetic program, or is it the accumulation of environmentally related compromises that reach a tipping point and cause autism? And I think that we're basing the treatment approach on the idea that there's a tipping point and that you can untip it. There's data showing, now this is a slide from Eric Kershane. The line here is the normal head circumference size. You see that at birth the kids can maybe even be on the small side. The head goes up 2 standard deviations on average in the first 15 to 18 months. That's after birth. Another study showing a similar kind of a trend. Even if it doesn't become frankly macrocephalic over the top, it still gets unusually large. And that this increase seems to be very fast in the beginning and then it tapers off through the lifespan. So definitely something's going on after birth. This is my own research, this little picture down here. I showed that the enlargement is in this outer zone, which is green, and that zone gets the white stuff, the myelin, substantially after birth, suggesting, just in a kind of a roundabout archaeological way, that something is going on that gets more intense as time goes on in development. Dr. Bowman and Dr. Kemper, who were strong advocates of this prenatal model, have recently commented in a paper last year that their finding that they reported 14 years ago, that autistic children will have larger, plumper cells than controls, while in similar or same regions the cells will be smaller and paler, represents an ongoing process, which they didn't identify, but that it suggests that there is ongoing biological change, and it's not just a wiring thing that gets a switch that gets flipped in utero. So all in all, we're talking it's not just prenatal, but there's plenty of evidence for an ongoing process. Now, critical question, and I've already said this, but I'm going to emphasize it, these ongoing processes, could they be treated? And that's where I want the field to go, beyond this room. Okay, now, another old model claim, genetically determined hardwired brain changes are obviously untreatable, but we're finding signs of ongoing physical illness in brains of people with autism. This is data from Vargas, Zimmerman, and Pardo at Hopkins, this one on the left, and this is activated microglia and astroglia, signs of ongoing inflammation in autistic brains, and they also measured inflammatory chemokines and cytokines. This is an abstract of signs of oxidative stress, lipid peroxidation, I'm going to say a little bit more about this, in the dendrites. And this is the little tippy tips of the ends of the cells which connect with the next cells and have a lot to do with how efficient neural transmission is. So inflammation is a body's way of protecting itself from infection and injury, but it can get out of control and dysregulated. And it's treatable by a lot of pharmaceutical as well as natural agents and over-the-counter agents, not all of which are appropriate for every type of inflammation. There are many types of inflammation. Oxidative stress, very, very, very simply put, and you'll hear a lot more about it, buildup of byproducts from demands on the body that exceed the reserves, and you can also treat this through a variety of agents. Just want to say this for those of you who are unfamiliar with these terms, and you'll hear a lot more about it from other people. Now, people assumed that these things weren't going on, particularly that there was no inflammations, but there was no inflammation. But the thing is, they didn't look in the brains with techniques which would show the inflammation. They looked grossly, it didn't look like multiple sclerosis. There were no big scars. They didn't find lymphocytes. But there are other ways of having inflammation, and when Vargas and Pardo and Zimmerman finally looked, they found it. MRI is not sensitive to that. This is well known to occur in Alzheimer's disease, and you can't pick that up with an MRI. It just doesn't see it. So this is a very important take-home lesson. What we know scientifically is only to the limits of the sensitivity of the instruments that we use. So anybody who gives you a definitive statement and doesn't take account of that maybe next year there'll be a new machine that sees better is not exercising the caution. We need to be humble about the limits of what we know. Now, this is another example where there are many studies showing that there's low brain perfusion, less blood flow in one or another region in autism. And most of the studies have been interested in showing, oh, if it's in the temporal lobes, it's about language. If it's in the parietal lobes, it's about integration. And actually, what's really interesting is they all show low perfusion, and none of the studies asked, what's going on with the tissue that the blood wouldn't be flowing well? They were basically asking psychology questions. Now, maybe it's a psychology consequence, but the cause of it is biological, and it needs to be investigated biologically. I keep coming back to that. So what are the significance of inflammation and oxidative stress findings? Well, it's ongoing and chronic. It's not just early changes. Environmental factors are well known to cause both inflammation and oxidative stress. These problems persist into adulthood. Those brains where they identified inflammation were of people who died at age 5 to age 44, and they all showed this. Autism now keeps company with a different class of neurological diseases that includes Alzheimer's, Parkinson's, and HIV, which have these kinds of changes. It suggests a biology, pathophysiology, with causes upstream and downstream cascading consequences. And also, as I keep saying, it's medically treatable potentially. And now, back to the slide I showed you from Margaret and Tom, I asked Dr. Pardo, when I first heard him present the inflammation data before it was published, if he thought that this phenomenon of the cells in a subtle way getting smaller over time could be a consequence of this kind of low-grade but chronic inflammation and oxidative stress. And it's plausible, but it hasn't been studied enough. So, something to think about. So, then this raises the question, the physical symptoms, is it just a coincidence or is it part of the underlying biology? Now, also, I'm going to be making a series of points right now just to hammer home what the implications are. Is the brain the sole and primary cause target of autism's causes, or it could be downstream? Could it be downstream of other things that are driving some of these changes in the brain? Could the inflammation and oxidative stress be because the brain is getting hit by things that are also hitting the body? So that means, is autism simply a mental disorder, or is that incomplete and is misleading? Genes, brain, and behavior may not define autism. They may participate in a larger network where there's a lot of other things going on, particularly physical symptoms such as gastrointestinal, immune, and biochemical abnormalities that are common in autism and that have certain patterns, and you'll hear a lot more about that in the whole conference. Now, this is where my work has been, the idea that I told you in the beginning that specific genes will affect specific brain regions and lead to specific behaviors. That's not what we find. That data is and not only that, we find changes all over the place. So this is my lab. I've shown this picture. This is a snow brain, anatomically accurate, gyri and sulci, and we have fun sometimes. And what we found, and if you look at the results of people looking for that brain region that will explain the behavior, those results are variable all over the place. There's no one region, there's no one type of change. Some areas are bigger, some areas are smaller. Some of them, like the corpus callosum, the part that connects the two hemispheres, tends to be smaller than expected. The whole size of the cerebellum, the volume, tends to be larger. But other than that, and the implications of those things are complicated. There's no one region found. The most single replicated finding in autism is that the brains tend to be larger. 97th percentile, above that, that means you're macro, that's big, cephalic is head. And that means only 3% are supposed to have that. About 20% of young autistic heads are macrocephalic, so that's way more than you would expect, and most are above the 50th percentile. So the distribution is shifted upward. It's atypical. It's not, there's no precedent for this in the literature. These are normal-looking brains if you just looked at them from across the room. There's a huge amount of literature on that, and it keeps growing. What does this mean? Could changes associated with brain enlargement lead to autistic behavioral syndrome? So is autism a network problem and not just a specific module broken causing a specific behavior, number one, different module broken causing different behavior, number two, gene A causing module one, anyway. It's not, doesn't seem to work like that. The people who still think that way, and they really do, because I've been talking with them, who really think that there's going to be language genes and repetitive behavior genes and social interaction genes, and when you get all three together, you're going to get autism. And they think that there are going to be genes for face processing, genes for social reward, genes for motor imitation, genes for memory, and that they're going to be tied up with specific brain areas that we know from other literature to be associated with autism. Well, there's two problems with this. First of all, there isn't a whole lot of evidence for most of these areas being unusual in autism. And second of all, the most prominent findings of large brain in particular, and also in large white matter, which don't make sense in this framework that a specific region should cause a specific behavior, are just left out. And I think this was a relatively innocent mistake. I think it just didn't occur to them, because their model focused them so much on this piece does that thing that they can't see an alternative framework. That's this rabbit-duck thing. They just can't make that shift from the rabbit to the duck. They can't do it. Now, another way of thinking about the brain works is connectivity. This is the poor guy in your brain taking the wires from one place and putting them in the other. Hopefully your brain works faster than that, but not always. And what we found in autism is that the connections between two regions here in controls, there's bigger gaps. The connections aren't as tightly coordinated in autistic brains from one area to another area. And here you see a series of pairwise connection, functional connectivity measures, and it's less in autism. Not in only one place, but all over the place. Barry Horowitz in 1987 at the NIH found this. He measured 800 some odd regions in the brain, and 70% of them had lower intercorrelation. So this is a finding that hasn't been pursued until recently. It's now really hot, but it's been around for almost 20 years. But Ben Horowitz didn't know what to do with it because it didn't fit into any framework, so he reported it and then it didn't go anywhere. There are models that have emerged of explaining the behaviors in autism that are things like weak central coherence or complex processing. You do the simple things well, you can't put the complex concepts together. These are models that are fit more with a big brain thing, and they suggest that the behaviors are most problematic in areas where you need the most coordination. That explains a lot of autism. I think there's more to it than that, which can be that certain chemicals irritate certain parts of the brain, which is a whole other thing, and I don't have slides on that, but I'm going to develop them for the future. So there are a couple of take-home points here. One of them is that do brain problems in autism have local addresses, or do they reflect problems in a larger network where the overall system dynamics are also compromised? And then the question, is there a connection between neuroinflammation or oxidative stress and network problems in autism? And that raises the really critical question on which this whole approach is based, which is the brain defective? Does it have an abnormal wiring diagram, or is the brain physically sick? And could this physical sickness lead to the network connectivity problems? And if you could treat this sickness, would the connectivity heal itself? And I think that that second possibility is what this approach is based on. The presumption that this is a hypothesis worth testing, and a lot of the ways that you can test it are pretty safe, and if you don't test it, it's hopeless, and if you do test it, you may have a shot, a pretty good shot in some cases. So now I want to say a little bit more about how some of the science or potential science around how this might work. The old assumption is that it's wiring, and the new model would be, could it be functional? Could metabolic problems be driving the behaviors? Now this is a slide on some of the ways that oxidative stress and inflammation can change brain function at the cellular level. Free radicals from oxidative stress can change energy production. There can be altered calcium metabolism, changing excitatory neurotransmitter function. Peroxidation, toxic mediators, transmitters are changed, and so forth. The point is that cellular changes driven by metabolism can be widespread, and they can impact the efficiency and coordination of brain functioning. So there are real ways of thinking about this, studying it, and treating it. Now, another thing, this is a hard to read slide, but the point of it is that when you have brain, a lot of people say that serotonin abnormalities drive autism, and it's been shown that when you deplete tryptophan, an amino acid which is out of which your body makes serotonin, behaviors get worse. Well, inflammation can deplete tryptophan because a pathway called the kynurenine pathway sucks off the tryptophan, and you don't have enough to make the serotonin. So you could get physical illness in the brain and the body, depleting the brain of the serotonin it needs, and that can make the behaviors worse. Now, John Rubinstein and Mike Merzenich from UC San Francisco wrote this paper in 2003 called Models of Autism, Increased Ratio of Excitation and Inhibition in Key Neural Systems. The idea is that there's too much excitation, that's the numerator, and not enough inhibition, that's the denominator. So you get, and I'll have another slide explaining that, you can get a lot of problems, which I'm going to say in the next slide, but what I want to say here is the part that I highlighted is what I think is one of the smartest sentences in the mainstream autism literature, which is that this increase in excitation inhibition ratio can be caused by combinatorial effects of multiple genetic and environmental variables that impinge on a different system, and it suggests therapy. So I'm going to show you that in the next couple of slides. Increased excitation and inhibition and reduced inhibition create a kind of a noisy cortex. If you have a signal going in and your cortex is very excited, you get an extreme overreaction. That can be consistent with the way kids can't stand various sensory stimuli. It can be consistent with poor coordination of timing. It can lower your seizure threshold, and we know that at least 30% of people with autism develop seizures, and depending on how you measure it, there's abnormal brainwave activity and a lot more. It can promote anxiety, it can interfere with sleep, and actually in the whole body, these changes can also cause problems like GI dysmotility and interfere with gastrointestinal function. So this is one of those mechanisms that I was talking about earlier, that you can have an increased ratio of excitation and inhibition. It'll mess up your gut and mess up your immune system, mess up your brain structure and function. And there are various genes that can set you up to be vulnerable to this, either lowering the denominator or increasing the numerator. And a lot of toxins can also do this. A lot of toxins create extra excitation in the brain. So if you have any kind of a genetic vulnerability, you'll need less toxic exposure to get kicked into this unstable and vulnerable state. And you can treat these things. So then, like as I keep reiterating, if there are contributors to brain problems that you can change, such as the toxins, the inflammation, and oxidative stress, then treating and improving these problems should improve brain function. Now, I'm going to zoom out from autism right now. Why are people sick? Why are there so many autistic kids? And many of you may also, there's a lot of people with autism, with kids with autism who are not feeling so well, and there's millions and millions of other people who are not feeling so well these days. This is a slide of toxins that you find in meconium. Now, meconium is the first bowel movement a baby takes when the baby is born. And this was a slide of 426 infants in Manila in the Philippines. They found lead, cadmium, mercury, chlordane, chlorpyrifos, diazinon, DDT, lindane, malathion, parathion, and pentachlorophenol. I pronounced all that. And that's in baby poop when you're born. And baby poop when you're born represents what you've been carrying around in your system the whole time you're in utero. So we are born polluted. Here's another study, body burden, the pollution in newborns. This came out more recently, just last year. $10,000 a kid is the first time cord blood was assessed for toxins. They looked for all kinds of toxins, and I won't read you the whole list, but they did find 287 chemicals in umbilical cord blood. 180 of them cause cancer, 217 are neurotoxins or brain toxins, and 208 cause birth defects or abnormal development in animals. So this is, again, what you're born with, and I think what we need to face right now is nobody is born clean anymore. That's over. And these chemicals may be at low levels, but they act together, and they can cause some of the changes, such as I was showing you, in how cells fire and signal and communicate and how organ systems function. If you want to learn more about this, you can Google some of the sites. Some of it's in your handout there. You can also Google how toxic is your bathroom. There's quite an article in there about untested chemicals in your bathroom products, like your cosmetics and so on. Skin Deep is another one about cosmetics. Just this data is there. Now, I want to give you a very oversimplified but somewhat useful framework here. It has to do with final common pathways. Our bodies don't have one pathophysiological mechanism for each chemical we're exposed to. We have certain ways that we detoxify and metabolize stressors. There are a lot of environmental inputs: chemicals, allergens, heavy metals, radiation, infection, drugs, toxins, stress, noise, maybe others. This all goes through the body's defense and coping systems: the gut, the lungs, the immune, detox. These systems can only take so much. They can get overwhelmed. If you have certain vulnerabilities, they're going to get overwhelmed faster. Your body reacts in several ways. One of them is it reacts generically. Generic reactions like inflammation and oxidative stress. They're not fully generic, but it's a way of starting to think about it. Then you also have your own genetic uniqueness, what time in development this happened, what specific characteristics of the agents, and what kind of state of health you're in, and so forth. So you get specific diseases, but these specific diseases can be triggered by just overwhelming you, below which the cells aren't dying, then it should be safe. And it's turning out that at extremely low levels, this is the one on the right, chemicals, they will not kill cells. They will hijack signaling mechanisms. They will act like hormones or other signaling molecules in the body, because your receptors in your cells won't be able to tell the difference between what chemicals your body would produce and a very similarly structured chemical that comes from outside. So at doses far below toxic levels, chemicals can do things like endocrine disruption. There are also synergies where two or more chemicals and combinations can have effects not seen with individual exposures. It's well known that in Parkinson's disease, people who are exposed to the two pesticides, paraquat and manab, will have a much higher risk of Parkinson's disease, but either one alone won't have that effect. Now, there was a study, many of you may know about Brick Township, which was an autism cluster which was supposed to have a high rate of autism. And people went in there and looked at the chemicals in the water supply and said, oh, none of these chemicals are above toxic threshold, so that's not it. Well, at Woods Hole Laboratory about a year ago, they published this paper taking 3 of those chemicals and studying them in combination pairwise, and all 3 at the same time. All 3 of them at the same time disrupted a cyclic AMP process very important to brain development. Either one, single or pairwise, that didn't happen. Now, there are 2 interesting things there. A decision was made that the chemicals in the water supply at Brick Township were safe based on only individual tests, whereas we now know that these things act in combination, number 1. And number 2, this was the first study of those chemicals that was ever done on a developing system. We know that the thresholds for developing organisms are very different than the thresholds in adults. So they made a decision on individual testing at adult thresholds for a system that's being affected by combinations of chemicals in early development. So basically what we're saying is that the way we've been testing and deciding what's safe needs to be rethought in a very, very deep way, because it's not really protecting us the way we need to be protected. And this is just a slide, I already showed you this, to say that these kinds of chemical things can mess up these kinds of signaling processes that I already showed you. So this is about autism, but it's also about more than autism. This is trends in chemical production, 1920 to 1980. 1920 wasn't a whole lot. Look at this, we have an awful lot of chemicals. Pesticide use more than doubled between 1964 and 1982. There are 85,000 chemicals in a federal inventory. There are 2,000 to 3,000 new chemicals introduced each year. At least 58,000 chemicals are not tested at all because they were grandfathered in before any testing was required. This is more increase in synthetic chemicals, somewhat less basic chemicals. This is insecticides that we carry around that are detectable in our urine. Up to nearly 100% of us will carry around some of these pesticides. I won't go through the whole list. It's just so you know that this is what our daily lives are modulated by. This is increase in childhood cancers, age 0 to 14, from 1971 to 2001. Leukemia is up 32%, ALL 53%, soft tissue cancers 32%, brain tumors up 63%, bone cancers up 32%, kidney cancers up 26%. This is hypospadias, a penis malformation where the opening of the penis is not at the tip but somewhere else. It's doubled. This is increase of refined corn consumption, pizza consumption, soft drink consumption, and sulfited potato consumption. So it's not just about, many people think that these things are no good for you, including a lot of people who are going to be teaching the docs and all of you that some of these substances are maybe part of the problem. This is Dr. Philip Landrigan at Mount Sinai, and he's talking about the new pediatric morbidity, a range of chronic conditions of complex and poorly defined origin that account for most hospitalizations and deaths among American children today. And I just finished attending on the wards a few months ago, and there's an awful lot of kids who come in with something, bad something. And then you run all the tests and you can't name it. And then what do? If you start thinking functionally the way we're trying to think here, you'll have more options, but that's a whole other talk. Now, we're all living downstream. This is Sandra Steingraber. She wrote a book called Living Downstream, an ecologist looks at cancer and the environment, and this is a book called Terminus Brain, the environmental threats to human intelligence. The idea that we were very clever to make all these chemicals, but now they're coming back and hitting our self-destruct butt because our metabolisms can't handle them. And it's also, I just want to point out that it's not just people who are sick and just randomly put in, this is sea level rise. I could give you environmental variables up one side and down the other. The planet is not stable. Even Scientific American pointed out that 1,360 scientists from 95 countries had a report a year ago. Ecosystem damage is so severe that we can no longer be confident that planet Earth can support human life for more than 2 generations. So many people who think that autism has an environmental contributor think of autistic kids as a canary in a coal mine and warning us that something is going on that's bigger than these kids and we should get a grip. And I've shown some of this. This is a joke. How many of you have read The Onion? It's at www.theonion.com. It's the funniest news source around. It's fake news, but Krugman from the New York Times calls it the preeminent source of news in the US today because somehow these days we're able to say things as jokes when we can't say them straight. And he points out that according to an EPA study, consumer product diversity, which is the red line, now exceeds biodiversity. Last year's introduction of dentine, ice, cinnamon gum, right on the heels of the extinction of the Carolina tufted hen, put product diversity on top for the first time. So today the Procter & Gamble subphylum alone outnumbers insects 2 to 1. But he says this is good news because these losses in biodiversity are more than offset by a corresponding rise in consumer product diversity. Though flora and fauna are dwindling, the spectrum of goods available to consumers is wider than at any time in planetary history, and we should all be happy about that. And this is my most cynical slide. And so while the end of the world scenario will be rife with unimaginable horrors, we believe that the pre-end period will be filled with unprecedented opportunities for profit. So I think that we need to understand that this is what we're dealing with, but the countervailing force is the insistence of everyday people that we really want a real quality of life. We want to get our kids better, and we don't have to go through this. It doesn't make sense. Now, this is a quotation, I'm not going to read the whole thing, attributed to Chief Seattle, and he said, "We are part of the earth and it is part of us, and whatever we do to the web, we do to ourselves. We do not weave the web of life. We are merely a strand in it. Whatever we do to the web, we do to ourselves. I've read this. Many of you have heard me say this before, but I'm going to read it again for those of you who haven't heard it, and also for those of you who've had. This is a reworking of that quotation. And it comes from a very important piece that I encourage you to read called The Age of Extinctions and the Environmental Health Movement. What will happen when a working woman comes to a realization, I'm sorry, I can't do this straight, but I'm going to do it anyway, that her own breast cancer, her husband's lymphoma, her brother's melanoma, her son's learning disability, his best friend's attention deficit disorder, her daughter's endometriosis, her niece's cleft palate, her cousin's chronic anxiety and panic disorder, her best friend's severe chemical sensitivity, her best friend's daughter's asthma, her uncle's infertility, her neighbor's son's testicular cancer, and her sister's daughter's childhood leukemia may form a pattern. What will happen when this working woman begins to understand that these new human pandemics that affect her family and her community directly may be profoundly connected to what is happening to the fish in the sea, the birds in the sky, and the animals of the earth? I believe this working woman will understand that the cancers and the infertilities of the fish, the disappearance of the frogs, the cleft palates of the mice, the shifts in gender orientations of the birds, the susceptibility to viruses and infections of the seals, the disappearance of the songbirds, that all this and much more may be telling us a story that is also our story. The story that the birds and the fish and the mice are telling us is the story of interbeing, the story that all life on Earth is truly, breathtakingly, concretely connected right now. That what we do to the mice of the field and the birds of the forest, we also ultimately do to ourselves and our families right now. I do not believe that we can hide from this story much longer. It is among the great stories of our time. So, I'm winding up. What do we do in this kind of evolutionarily unprecedented crisis? How do we respond thoughtfully? Well, on an individual basis, I've already said, and you'll hear over and over again, that the way our bodies are hit by environmental toxins, infections, stressors, may teach us about what we can treat to get better. And that's what we're trying to do here. Final common pathways. The liver has phase 1 and phase 2 detoxification. The enzymes in the liver are supported by nutrients. Riboflavin, niacin, pyridoxine, folic acid. A lot of these things are what are included in what you will be taught about how you can help your kids. Nutrient support. Nutrients are cofactors for biochemical reactions in the body. It's just very important to remember that they're the building blocks of our body's metabolism. Glutathione. Jill James wrote a paper showing that glutathione, one of the key detox chemicals in the body, can be protective in the setting of exposure to thimerosal. Interestingly, the Pentagon has a patch, a glutathione patch. They got burned, I guess, by Gulf War syndrome. People got, 20-some-odd vaccines, depleted uranium, and all kinds of chemical and biowarfare agents in a short period. Anybody with a genetic vulnerability would have a problem. So now they're going to send people out into that nightmare with glutathione patches. So they get it, that there's nutrient support. I wish they would get it that they shouldn't send people out into those situations. That would be a good idea. Now, this is, I've already said some of this, but how many chemicals are tested? Of the chemicals that are produced above 1 million pounds per year, there's some testing data on 21.4 for overall development, not brain, no development testing on 78.2, and only 12 are tested for neurodevelopmental toxicity, the impact on the developing nervous system. That test is not required. So we have no clue what 85,000 plus chemicals are doing to develop. We have no idea. Now, this is another joke from The Onion. The EPA used to drop the E and the P from its name. Washington, D.C. Days after unveiling new power plant pollution regulations that rely on an industry-favored market trading approach to cutting mercury emissions, EPA acting administrator Stephen Johnson announced that the agency will remove the E and the P from its name. We're not really environmental anymore, and we really aren't protecting anything, Johnson said. The agency is a name that reflects our current agenda and encapsulates our new function as a government-funded body devoted to handling documents, scheduling meetings, and fielding phone calls. The change comes on the heels of the Department of Health and Human Services' January decision to shorten its name to the Department of Services. Now, how are they going to test 85,000 chemicals? It's bad enough they haven't tested them one at a time. If you tested just the top 3,000 in combinations of 3, that's 85 billion tests. No, it's impossible. We will never have precise knowledge of what on earth is going on right now, walking around with hundreds of chemicals that we just, of the ones we measure, not counting the ones that weren't in the profiles. We have no idea. So what does that mean for evidence-based medicine? What does that mean for evidence-based medicine when we're told that we don't have double-blind placebo-controlled studies about the interventions we're doing? Where are the double-blind placebo-controlled studies on those 85,000 chemicals? Where are the double-blind placebo-controlled studies on the 85 billion possible combinations of the top 3,000? I mean, we don't know. So we have to use our judgment. We have to think differently about what we're doing. And that's why I called this talk not evidence, but plausibility. We're in a crisis and we need to use our heads and use our judgment. So I just want to talk about the kinds of things that would be really helpful to do, to have happen, to make that possible. We need research. I need my autism research colleagues to measure wet stuff. They're not doing that. They're measuring genes, but they're not measuring chemicals. They're not measuring, for the most part, immune. They're not looking in the wet stuff for metabolic biomarkers. We need to look for subgroups so we can understand that different kids respond in different ways. We need methods so we can stop washing out response of things that work for some kids and don't work for others, and you average it all together and the response goes away, and then you say, oh, that stuff doesn't work and it's just a bunch of kooks. We need to have an individualized approach to what we're doing, and we need to understand that the behaviors that we see are a final common pathway. Now, this is my last complicated slide, and I know I am rushing. This is my pathogenesis mechanisms phenotype thing I've been going over. Is there one thing that causes autism, or is it an interaction of a lot of things at the pathogenesis level? We don't know. I suspect that there's a lot of mutual potentiation. Just for example, mercury and PCBs together make each other a lot worse. And we're all exposed to PCBs and not just mercury. Is there one particular kind of molecular or cellular mechanism, or are there a variety? Is it just neuroinflammation, oxidative stress, or are there a variety of metabolic changes? My sense is that there are a lot of variability in here, although there may be some big subgroups. As long as you mess up processing in the brain, as long as you mess up that network in some kind of way in a specific developmental window, you can wind up with something that would, at the behavioral level, qualify as autism. That makes research very complicated. It's not like you got tuberculosis, you got the bug, you got the disease. It's a very different ballgame. 21st century medicine about chronic, environmentally modulated diseases is very complicated, and we have to stop using 19th and 20th century methods of research to study something that's a whole hell of a lot more complicated than that. And I think that's critical. And this is not just about autism, this is about the epidemics of asthma and diabetes and cancer and on and on and on and on. It's about all of that. So individualized treatment is a challenge of the 21st century. For autism and for other things, we need to have detection. We need to have an emphasis on how do we find out who's at risk so that we can modify vaccines and other things that we do, foods that we give, nutrients that we give. How do we biologically test for autism and for risk of autism? What would be effective prevention? What do we avoid and what do we support? And how do we really optimize recovery, reversal and recovery? How do we treat aggressively? And we should treat with the full expectation that every child should have a chance, a real shot at recovery, and there's no child left behind. Many of my colleagues can't handle this for the following reason, and I'm going to read this to you, to cling to a purely genetic explanation, which many of them still do. I had somebody tell me a month and a half ago, 98% genetic. To cling to a purely genetic explanation of autism is a desperate attempt to maintain the illusion that one lives in a comfortable and rational world where new chemicals and technologies always mean progress, experts are always objective and thorough, corporations are honest, and authorities can be trusted. That human actions rather than genetics might be responsible for compromising the health of a significant proportion of a whole generation is so painful as to be unthinkable. Now, this is just some of the selection of nightmare scenarios that I've shown you in the course of my discussion. I think the scientific establishment needs to do step one from Alcoholics Anonymous, which is admit that you have a problem. If you don't admit that they have a problem, you go down a lot of garden paths, and there are a lot of garden paths to go down. We could focus on really leveraging what we already know to really help a lot of people if we admitted that we have a problem. Step two is really crisis-oriented strategic research and interventions to leverage and maximize our chances at getting a grip on this and on a lot of the huge problems that are facing the whole population, species, and the planet, so that we can heal our children, heal ourselves, and heal our planet. Thank you.
Wonderful.